So hello, everybody. Thank you for coming to the second Variant Effect Seminar Series seminar. So to go over some quick housekeeping things. So during the session, you can post your questions to the speakers anytime in the Q&A thing. And so the speakers are going to talk for 20 minutes, and then there will be a 10-minute Q&A session moderated by Diego and myself. Another thing is that the AVE Alliance now has a Twitter account, so you can follow us on Twitter at Variant Effects. And then also, if you're not already, you can become a member of AVE on Slack, and we have a dedicated channel there to discuss these seminar series. And so today, we have talks from Jonas Koeppel, who is a PhD student in Leopold Parts Lab, and he's going to be talking about the determinants of efficiency of writing small sequences into the genome using prime editing. And then second up, we have Isaac Gia, who's talking about a deep mutational scan he's done following up on his MSH2 screen he had done previously, where he's now looking at MLH1. So first, we're going to have Jonas speaking. And so I know a lot of the community is excited about using prime editing for variant effect analysis, but those of us who have hands-on experience using it, we have all found that it isn't always as easy as it seems. And so there's still a lot of room for optimization here. And Jonas is doing some really exciting work trying to find out specifically what does or does not allow efficient prime editing, specifically with respect to writing small sequences into the genome. And so if you have anything beyond the questions that are happening in the Q&A session, you can contact Jonas at either of these two links here. So with that, if you want to take it away, Jonas.
All right, thank you for the introduction. Everything looks okay with sharing the screen?
Looks good.
Perfect. All right, yes, as Stephen already said, so I'll talk about the determinants of efficiency for writing small sequences into the genome using prime editing. And I think because it's a variant effect seminar series, most work will be done on changing individual amino acids. But I guess I'm here to also convince you that there are a lot of very interesting things we can do if we can insert short sequences. And this is one of the great things about prime editing, that it gives you this flexibility that you can do small deletions, point mutations, or insert sequences. And so just a few use cases. For example, in biochemistry, we could tag proteins or mark them with different localization sequences, or label them fluorescently using short GFP constructs. That could also be then a readout for a different variant screen. Then if we think about modeling structure variants, we could also insert recombinase sites, for example, and then flip around sequences or delete them, or generate landing pads for integrating bigger sequences. In terms of functional genomics, we can, for example, do screens and insert stop codons and thereby knock out genes without causing double-strand breaks. Probably most importantly, there are many pathogenic deletions that are associated with some sort of disease, and we could, in theory, use prime editing and the ability to write small sequences back into the genome to fix those mutations. The tool here is prime editing. So just a quick summary of how it works. We have a nicking Cas9, which can make a single-stranded DNA break at the target site specified by the guide RNA. And then in case of prime editing, the guide RNA is extended at the 3' end. And so that can then encode for an edit that we would like to incorporate into the genome. And in our case, we were very interested in small insertions. And this is then read by a reverse transcriptase, and repair processes, and we also talked briefly about those, can then fix this reverse transcribed sequence into the genome later. So what did we do? We designed a library that contains around 2,600 sequences. And what type of sequences do we have? We have utility sequences. So these include all sorts of tags, recombinase sites, and different, just whatever we thought might be useful for people to insert into the genome. Then we just took the entire eukaryotic linear motif library of small sequences that are defined to have some sort of function, degrons, localization sequences, and so on. And we plugged them into our library. And then we are also specifically interested in how secondary structure affects insertion. So we have another sublibrary that contains around 400 sequences that have strong structure. We decided to go for 4 target sites. Those are target sites that have already been characterized in the original prime editing paper. And then we chose 2 cell lines. One, the HEC293 T cells, because we thought these would have a very high likelihood of success, and they have been extensively tested for prime editing. And also the hub1 cells, because we thought if we are interested in generating variants, it's maybe good to work in a haploid background. And there we did 2 of the target sites. Just to show you roughly what's in the library, so this is the different types of sequences stratified by length. So you see that we have many sequences, around 20 nucleotides, some that are very short, including all singlets, doublets, and triplets. And then we have sequences up to 69 nucleotides. And they have a kind of range of GC contents. So with that, we designed a screen in order to insert all these sequences into those target sites and cell lines, and then follow up which sequences inserted more efficiently than other sequences to hopefully generate some understanding of which sequences work well with prime editing. The way we did it is we first cloned this pegRNA library into a lentiviral backbone with a blastocidin marker, so we could infect cells and select for cells that have the library. Then we transfected in prime editor 2 and selected cells that got the prime editor using puromycin. And then 5 days after the start of the experiment, we harvested the cells and extracted the DNA. And then we sequenced 2 amplicons. One amplicon is the target site, and the other amplicon is the pegRNAs. So basically, we can make a ratio to see how many cells that had a given pegRNA also inserted a given sequence at the target site. And Ellen helped me a lot with those screens. Actually, I think she did more than me. She's an amazing research assistant in our lab. So first of all, here are the average editing efficiencies in the 2 cell lines and across the different target sites. So you can see that there's quite a range of editing activity. Our best rates were observed in the HEC3 locus in HEC293 T cells. Each dot here represents a replicate. We did 3 biological replicates. And then you see that there's also some level of indels, but it's relatively low. This is actually, I think, an overestimation, the way we are calling them. And you see that the HEC3 site, for example, works less well in hub1 cells, and all the other sites work less well than the HEC3 site. And we'll also talk a bit about target site differences in a moment. Yes, so we were interested in how consistent are the things we see in terms of how consistent, if one sequence is inserted well in one target site, how often do we see it inserted well in another target site? So first we looked at biological replicates. So between the triplicates, you see that we get quite good correlations, which means that insertion efficiency is reproducible. A sequence that inserts well in one experiment will also insert well in another experiment. And then, as you can see, that if we look at the same target site in 2 different cell lines, actually I can also show you the plots. So this is replicate correlation. This is the percent insertion in one replicate, and this is the percent insertion in another replicate, and you can see it's very nicely aligned. Then if we look at the same target site in 2 different cell lines, you see that the correlation overall is still quite good. However, there's this island here in the scatter plot, and we talk about this in a moment. So the correlation gets worse compared to replicates. Then if we instead look at the same cell line, but target 2 different sites, the correlation is slightly worse than looking at different cell lines in the same target. And the correlation is lowest if we both change the cell line and the target site. With that, we were now interested to understand which features determine how efficient any given sequence can be inserted using prime editing. So let's first look at the size of the insertions. And this is a diagram that plots the percentage of cells that have a given insertion versus the nucleotide length of the insertion sequence. And you can see there's this interesting pattern that short sequences are inserted quite well with an average of around 20%. And then we have this dip. So sequences around 10 nucleotides seem to be inserted poorly. And then we have a second optimum here that is around 18 nucleotides, and those sequences insert quite well. And then the insertion rate keeps dropping as we go longer. So this was, what I showed you was one locus in one cell line. If we now expand to all 4 loci, you see that the pattern is more or less preserved, even though we have an order of magnitude difference in absolute insertion efficiency, but we still see this pattern of short sequences are inserted well, those middle sequences are inserted quite well, and then it drops off as sequences get longer. But how if we compare across different cell lines? So this is again showing the HEC3 locus. And we were quite surprised to find that there appears to be a difference between the HEC293 T cells and the HAP1 cells. And the difference seems to be specific to those short sequences. So what you can see is in 293 T cells, the short sequences insert quite well. But in HAP1 cells, the short sequences don't insert well. However, for the rest, it looks similar. And we can also plot the insertion frequencies in the 2 cell types. And you can see that they're quite correlated overall. However, you can see the same phenomenon that the short sequences really stick out as being inserted much better in the HEC293 T cells. And you can also see that the correlation for the entire plot is worse than if we look at the correlation for each of the sequence length bins indicated here and in different colors. So there must be something different between the 2 cell lines that specifically affects shorter sequences. And we reached out to some collaborators who were in the DNA repair field because we thought maybe this connected to DNA damage repair. And they suggested that we should look at mismatch repair and specifically MSH2 and MSH6. And also it was quite intriguing that I learned that the HEC293 T cells that we're using are partly mismatch repair deficient because they have a hypermethylation in the MLH1 promoter, so they don't express much of MLH1 versus the HAP1 cells that are proficient in mismatch repair. And then at right about the same time, there was the conference, Cold Spring Harbor conference, where Peter Chen from David Liu's group talked about improvements of prime editing, and they figured out that mismatch repair inhibits prime editing, and this was recently published. So we knew we had to do this experiment in a mismatch repair deficient HAP1 cell to see whether that caused the difference. And so we ordered a cell clone. Again, to orient you, we have the HEC293 T cells and we have the HAP1 cells. And now we also have data for HAP1 cells that are knockout for MLH1, an important component of the mismatch repair pathway. And you can see that the short sequences insert very, very well in this cell line. And also, if you look at absolute numbers, it's I think around 40-fold higher for the short sequences compared to the HAP1 cells that are wild type in MLH1. And overall, it's around, I believe, 6-fold higher, the editing rate in those cells. So with this, I think we understand what causes the differences between these length to insertion patterns. And also we can again look at the scatter plots. We have this island here when we compare the wild type cells. And then if we take the HAP1 cells that are MLH1 knockout, you can see how all the sequences align very, very well between the 2 cell lines. And in fact, the correlation is as good as the correlation between replicates. And then we can also compare the HAP1 cells that are knockout for MLH1 to the wild type HAP1 cells. And now we can see that, again, the short sequences insert much better in the MLH1 knockout cells. So a short summary of the insert size effects. We observe that if we have mismatch repair deficient cells, we can very efficiently insert short sequences and moderately efficiently insert sequences between 18 and 21 nucleotides. But in comparison, if we have mismatch repair proficient cells, we cannot insert those short sequences very efficiently. With that, we can move on to what actually the composition of the insert sequence, how that affects prime editing efficiency. And so first of all, I would like to show you the effect of GC content. So what you see here, again, is every dot is a PEG RNA, an insertion. And then if it inserts better than other sequences of the similar length, then it's positive. And if it inserts worse than other sequences of a similar length, it's negative. And you can see that in all target sites and cell lines, there's a positive correlation with GC content, which means that insert sequences with higher GC content insert better. And we can also stratify this by nucleotides. So this is the GC content again. You see that all cell lines and targets insert better with higher GC content sequences. And then if we look at each of the individual nucleotides, it seems like having A is quite bad for insertion rates, having T is quite bad, having G is a bit of a mixed bag, but then having specifically cytosines seems to be very beneficial to insert sequences well using prime editing. And I can show you this for one specific example, which is the His6 tag. So it's a common tag used for protein purification, and it consists of 6 times the His6 codon. And there are 2 versions of the histidine codon, CAT and CAC. And then you could target the forward or the reverse strand. And if you target the reverse strand, you could use the reverse codons ATG and GTG. And you can see first, if we just look at the forward strand, you can see that the version of the His6 tag that uses the highest GC content and specifically the highest content of cytosines inserts best in HEC3 in the HEC3 site and HEC293 T cells, but it's about the same in all of the other targets. And the one that minimizes the GC content and the cytosine content inserts quite poorly. And then if you look at the reverse strand, actually all of them insert worse than the forward strand. And that's consistent with this effect of having high cytosine is good for insertion. But even there, you can see that having the version that maximizes GC still inserts slightly better than the other versions. But this basically gives you a rule of thumb of how to optimize insert sequences for prime editing. And it looks like if you pick cytosine-rich codons, you're usually good to go. And yeah, this is also just showing the codon table of all triplets. And you can see that that all the triplets that start with C or have C as a second base work quite well. And then this section where the 2 intersect works best versus the ones with T work poorly. Although this is a bit of a mixed bag with the codons for different target sites, but the His6 tags are quite consistent. Then what other things matter? We also saw, I told you before that we're interested in secondary structure, and we see there is a correlation between secondary structure strength and insertion efficiency. We used VienaFold, it's an online tool to calculate the free energy of a secondary structure at 37 degrees for each of our pegRNA inserts. And you can see that negative values here correspond to a pegRNA insert forming a strong structure. And so you can see that the pegRNAs that form strong structure are inserted better than the pegRNAs that form weaker structures. And this is quite consistent. So what I'm showing you now, each dot is a correlation for one experiment between secondary structure and insertion efficiency. And so you see it's negative because these free energy values are negative for strong structures. But you can see having structure is almost always good for the 30 nucleotide inserts to insert well. And our 20 nucleotide inserts were also designed to contain a lot of them that have strong structure, and we see the same thing here. However, the correlation is much weaker if we look at the entire library, because many sequences in the library don't form secondary structure. And so we thought that maybe what matters is not just the structure that the insert can form on its own, but maybe in combination with this entire 3' extension of the prime editor. And then you see those are the black dots. Again, each dot is a correlation in one experiment. And here you can see that it works much better for the entire library, where many of the inserts alone don't form structure, but form structure in context of this 3' extension. So that was some of the insertion sequence effects we found. And then finally, I would talk a few minutes about CRISPR system effects. And so first of all, it is known that the occurrence of many Ts acts as a terminator motif for the polymerase 3 that expresses the PEG RNA. So we wondered whether having that in the insert of that we're trying to insert would decrease editing rates. And as you can see, that's a very clear effect that whenever the inserts have many of those consecutive Ts, we see lower editing efficiency. And out of our library of around 2,700 sequences, there were 23 that were not detected in, 24 that were not detected in any of our screens. And of the 24 that weren't discovered, 23 had at least one of those terminator motifs. Then the original prime editing paper proposed two strategies. One strategy is dubbed PE2, which just uses a prime editor. And then the other strategy is dubbed PE3 that also uses a nicking guide RNA that nicks some 50 base pairs away from the prime editing site and nicks the opposing strand to facilitate removal of that strand and keeping the edited strand. And that was shown to increase editing efficiency. And we see that it also increases editing efficiency for us. It was also proposed to increase indel rates. We don't see too many indels with PE3, but definitely more than using PE2. But we were then, of course, wondering whether the factors that determined if a sequence inserts well using PE2 also affects, also hold up for PE3. And thankfully, we can see that they're very well correlated, basically as correlated as replicates. Although, again, we have quite a bit of a difference in absolute insertion rates, but kind of the relative ones between sequences is well preserved. So to summarize, these effect parts, we see the size effects, we see there's the effect of GC content and structure, we see the effect of terminator motifs, and we did not see differences between P2 and P3. So we thought of putting all those together in a computational model to predict insertion efficiencies. This was mainly work done by Juliane Vella, another PhD student in our lab. And so she tried a range of different models and found that gradient boosted trees work reasonably well for this problem. So what I show you here then is the prediction on the y-axis and the measured insertion efficiencies. And you can see that there's quite a nice correlation between what the computational model predicts versus what we measured in our screens. So this is combining data from all screens, so all target sites and all cell lines. And also we can turn to SHAP values to get a glimpse into what this model learned. And you can see that, for example, it learned that length matters. So if the dots are blue, it means length is low, and it means that low length has a positive impact on the insertion efficiency. Then you can see that secondary structure, if it's low, it means it has a lot of structure. Again, that's positively correlated. And in the same way, high C content is correlated with insertion efficiency. So it's quite consistent with what we've seen just by plotting the things against each other. And then finally, how well does it generalize? So what we did here is we trained the model and withhold data from one target site and then trained it on all the other ones and looked how well it would perform. And you can see that it generally performs pretty well, even if a target site has never been seen before. And finally, returning back to our HIS6 tag that we talked about before, we saw those measurements and we can see how the predictions and the measurements line up. And you can see that while the model is quite good at predicting which sequence would work best. So this is the predicted insertion efficiency. This is the measured insertion efficiency. And you can see that if you just go by ranks, if you pick the top sequence from the model, you're getting the sequence that inserts most efficiently. However, the magnitude is still a little bit off between prediction and measurement. But this makes us quite confident that the model would be a good tool for picking codon variants of whatever sequence we would like to insert. And with that, the final summary, we did a screen of around 2,600 sequences with prime editing into 4 target sites and in 3 different cell lines. We found that length, GC content, specifically C content, the structure and terminator motifs matter for insertion efficiency. And we could put those into a model that predicts insertion rate reasonably well. And then in the future, there's still some discrepancies between target sites. You've seen that the model performs less well on a different target site than if we test it on the same target site. So there's still some differences that we have not captured. And then we've done everything with one PBS and reverse transcriptase template length. So there might be differences when we vary those. And also in the meantime, there were quite a few improvements to the prime editor system, which might be worth testing with the library. And with that, I'd like to acknowledge Jana, who did the modeling part, and Ellen, who did a lot of the screens. So yeah, we are all joint first authors on this project. Then Leo, who is supervising it, and then Fabio, Ananth, and many other people who kind of contributed with discussions and so on. Yeah, we'd be happy to answer some questions.
Okay, thank you, Jonas. Good job. So we already have a few questions here. I'll start with one from Florence, where they've asked, why do you think there's a dip in insertion efficiency at about 10 base pairs? So you've shown that mismatch repair is probably what's explaining the shorter insertions, and then you see another peak around 20 base pairs. Do you have any speculation as to what DNA repair pathway or what is driving that there?
Yes, maybe we can speculate together. So short answer is I don't know, but I think there's some somewhat interesting things in here. So if you notice, I'm not sure if I'm crazy, but there's like quite a consistent kind of slightly higher rate at just a little over 40 as well. I haven't checked if maybe some of those sequences, they're very sparse, as you can see, might have some specific features that make them insert more. So this might just be chance, but it seems somewhat reproducible across most of the target sites. So maybe there's something about a turn of single-stranded RNA or something like this that make this length insert well, but I'm not sure. And I think there would be very interesting experiment for the PE4 and PE5. What they did was this repair-seq where they knocked out with CRISPR inhibition a range of enzymes involved in DNA damage repair, and then looked at which knockouts would increase editing rates. But they tested this on, I believe, single nucleotide substitutions. So it might be cool if they would, or someone would try similar experiments using insertions of different sizes, because maybe it's different repair enzymes that would be responsible for those. And so maybe there would be a different avenue to maximize editing efficiency of these longer ones.
Right. And so there's also a question about what software you use to calculate the secondary structure of the 3' extension. Yes,
so that's VienaFold. You can either use it directly as a web version. So if you just type in VienaFold, what we used was RNA-fold of the VienaFold suite. And then they also have it as a command line tool, which we use to calculate it for the sequences in the library.
Right. And so there's one more question here, and it's earlier in your presentation, you showed just the insertion efficiency at the different loci you tested, and it was clear that the HEC3 locus had much higher editing efficiency than others. And I know this is a choice locus for most genome editors, but the questioner wanted to ask if you have any speculation as to why it is so much more efficient, like it's markedly more efficient than any of the other sites you tested, if you looked at anything there.
There's something about the HEC3 locus that just seems to work really well. I've tested a few things as well. And so for one, there's a very good paper by the Kim lab that characterizes a model called DPE that looks at many different target sites, because this one is quite limited in scope in terms of target sites. And I think there are a bunch of things like, so for one, how good of a guide RNA this matters. Then the GC content of the primer binding site matters. So there are a lot of features that determine whether one site is good or not. And I haven't run it for those sites, but I would assume that maybe the HEC3 site would also score well in their model. And then there's a bit of a weird one, but the HEC3 site has a SNP in the reverse transcriptase template that we're using. And we're using a 34 nucleotide reverse transcriptase template, which we picked because this was the original paper used for their LOXP insertion into the HEC3 site. And we tested a few lengths in the HEC3 site and noticed that 34 works best. However, I think for almost all other sites, it's too long. But because the SNP, I think maybe that just kind of somehow affects the binding energies that it makes 34 work very well, but it's suboptimal for the others, just two speculations.
All right. Well, it doesn't look like there's any more questions. Thanks again for the great presentation.